from San Jose, California, and brought to you by Think Tank Learning. Welcome to Meet the Experts. This is a forum in which we pick the brains of experts and professionals from various industries. We give students inspiration by showing them the academic journeys that they can imitate as they prepare to become experts themselves.
Welcome to Meet the Experts. I'm David Nguyen, and today we're going to talk about integrated bioimaging. That is, we will peek into the inner workings of molecular complexes, organelles, cells, and tissues. Our special guest today is at the forefront of electron microscopy to study things ranging from bacterial cells to human cells. He is Manfred Auer. Manfred Auer is a staff scientist at Lawrence Berkeley National Laboratory, otherwise known as LBNL or just LBL. Manfred Auer is one of the world's leading experts when it comes to applying electron microscopy to understanding biological problems. At LBNL, he is the head of the Cellular and Tissue Imaging Department, which is in the Molecular Biophysics and Integrated Bioimaging Division at Lawrence Berkeley National Laboratory. Furthermore, he is the Director of Physical Analysis at the Joint Bioenergy Institute. Dr. Auer, welcome to Meet the Experts.
Hi, David. Thanks for having me.
Wonderful. Thanks for being here. I just want to say to our audience that while I was a grad student and a postdoc at LBNL, I was able to hear Dr. Auer's presentations, interact with his lab members and with himself. And one thing that really stuck out to me about Dr. Auer was his ability to put teams together, to lead teams, to gather people's opinions, expertise, and really guide us in a grand vision of what it is that our science can do. And also through interacting with him and his lab members, he certainly has a high standard for integrity and the quality of the research that comes out of his lab. So I'm really happy that he accepted the invitation to join us today. I think you will really enjoy and get a lot out of all that he's going to share with us today. Okay, so with that, let's start with our first question here that I want to ask you. That is, what is electron microscopy?
Okay, so electron microscopy is not unlike light microscopy, where you take a sample and you penetrate it with, in light microscopy, light, and in electron microscopy, electrons. And you observe the refracted light that has interacted with the specimen, and by bundling back with the lens the diffracted light, you get an image. And so one of the major differences between light microscopy and electron microscopy is that electron microscopy works at the level of molecules, and so we can literally look at proteins, and we can literally look at the inner workings of cells, whereas the light microscopy, at least in classical light microscopy, is sort of limited to organelles, so in terms of resolution. One of the big problems with electron microscopy is that since electrons interact very much more intensely with any material than light, our samples need to be very, very thin. Typically, we want them to be as thin as half a micron, or even in our case, not bigger than about 100 nanometers. And that means we have to take our samples and we have to cut it in very, very thin pieces. And the other problem that we have, since the electrons are interacting so strongly with matter, we need to operate the microscope in a vacuum. Otherwise, the air would just absorb the electrons. That means the challenge that we have is that our samples need to be very thin, and they need to be working in a vacuum. So biological material cut very thin in a vacuum would dry out immediately. And then there are sort of various ways how to overcome that. One of them is freezing, and that is called cryo-electron microscopy. Or another way is to embed the sample into a resin, which allows you to preserve the sample and its original organization, but you now have a very hard resin. If you ever try to cut liverwurst, you know that you can't cut it very thin, but a salami you can cut very thin. And the difference is because the salami is very hard. And likewise, we need something that is very hard. And that can cause some issues with sample preparation, so it's a tedious technique. But one of the fundamental problems that it has is that we can only look at very small parts of the cell at any given time. And that is something I'll come back to later, has recently overcome with a sort of a new technique that allows us to go through entire tissues at a very high resolution.
Wow. So, I mean, what you described sounds very technical. And
yeah.
how much training does it take somebody to be able to do something like that?
Well, I'd be happy to answer that. But also, I think one thing it might be useful to show you what electron microscopy can do. And maybe that will help people understand why it's such an interesting and cool technique. But coming back to your question, I'll do that in a second. But coming back to your question of how much training is needed, it is something that does take a bit of time to learn. So when we have students joining our lab, and the students are usually attracted by the visual aspect of our work. It's just fun to peek into cells and see what they look like. It usually takes them at least or so half a year to get good at all the various aspects of this technique. So it takes about a half a year of training, and then after that, it's just a lot of fun. So if you want, I can share with you a little presentation.
Yes, please.
So let me see if I get that right. Did I? Oh yeah, start screen share. So hopefully, yeah. So that's worked. So let me actually go up here, and I just wanted to say a couple of things. One of them is what we really are trying to understand is how life works, and life works at the various levels of detail and complexity. And so we need to look at this at different scales, and we also need to look at this at sort of different levels of resolution. And we also need to there are different aspects of the system, architecture, composition, that we're interested in. And I would submit here that people know about omics, proteomics, genomics, which are typically bulk analysis methods that give you a parts list. And as you see right here, a parts list. But from that, it's kind of hard to actually guess that this is supposed to be an airplane. And what you need is to see how all the parts fit together. And imaging can be very helpful to bring the parts together in order to understand that this is flying. And the other point I want to make is that while we understand a lot about genes and genomes, and we have a good understanding of proteins, and we even have a reasonable understanding how protein work together to make these macromolecular complexes like cellular machines. We don't really have a good idea how all those machines work together to build up a cell, let alone how different cells make communities, be that microbial communities or being that a tissue. And that's really where the fun lies as far as I'm concerned. Just want to point out that we're spanning about 7 orders of magnitude, all the way from macromolecular proteins, like proteins, macromolecular machines, even smaller molecules, all the way up to entire organisms, like a zebrafish larvae, for example. So we're going from 1 nanometer, 2 nanometer, all the way up to millimeters. And just to put that in perspective, what that means, 7 order of magnitude. If you think about as your smallest unit, 1 foot, then it would take basically this order of magnitude would get you all the way from California all the way to New York. And trying to understand something like the United States at the foot by foot level, you can probably see the complexity that we're dealing with. And the question, what is the best approach to do that? There are a variety of different techniques that we can use. They're all very specialized. But the real challenge is to bring them all together. That's one of the big themes of my lab. So we all used, by the way, you can see the screen, right?
Mm-hmm.
Okay, so you all used this nice little cartoons of what cells sort of looked like. But then you actually look what cells really look like, and you actually realize it's very complicated. There is so much information in there. For example, I can point out those are mitochondria right here. Those are vesicles. You can see microtubules, for example, microtubules right here. And that's just 100 nanometer slice through a cell. So that's about 100th of the thickness of a cell. And so you can see the challenge is the complexity. So we'd like to capture all of this, and we'd like to quantify all of this. And that's where my lab is trying to go towards soon. Things become even more complicated if you look into a tissue. I should say that we're looking at here at fairly high resolution at an area that is about 10 times bigger than a cell. And when you can see is there's a lot of detail in there, but also what you should be able to see, if I just roll it back one more time here, is that you have a high level of heterogeneity in there. None of this cell nuclei looks like any of the other cell nuclei. So the ability to deal with heterogeneity in biological data is going to be a big problem. So then, when you look at this here, every single one of those little tiles here that you can barely make out here is a 2000 by 2000 pixel image. That's like 4 megapixel, about what your camera on your iPhone will sort of give you. And we put thousands of those together to build up this entire organism. And it's just mind-blowing, the ability to go in there and trying to make sense of what we see. It's very, very complicated. This data set is actually not shown at its full resolution because there is no computer that is powerful enough to actually display the data that we have taken on here. So those are the challenges that we sort of have. And another technique that's sort of coming in that's helping us is X-ray microscopy, which is kind of like a CAT scan in terms of medical imaging. And that allows us to look at the organ, and it also looks us at the tissue level here, and it allows us to see cells and see organelles. At which point we switch over to a different technique, and that allows us to look at individual cells and to look at them in great detail. This is sort of the very forefront of electron microscopy imaging. This is a dream come true for us, the ability not only look at 100 nanometer worth of a cell, but now going through entire cells. And that changes everything about what you thought you knew about the cells.
And can I ask you a question? What you just showed us, was that individual images then stacked, or is it like?
That's right. So we going. Sorry, I always assume people understand what I'm looking at, but of course not. What you actually see here is you see, for example, mitochondria. You see a nucleus coming in a minute. There is right here you have endoplasmatic reticulum, and you look at this at 5 nanometer intervals. So we're going through a 3-dimensional volume, 5 nanometers at the time. And it's not that anything is moving, it's just that you're following a 3-dimensional object slice by slice. And here's now the nucleus sort of coming in. And you see that real cells look very different than the cartoons that we make of them. And then when you take those mitochondria, for example, and the endoplasmatic reticulum, you can manually segment those. We can take them out, pick them out, extract those features. And that actually took many weeks. Then we can get this very elaborate representations where in gold you have the mitochondria, and in this salmon color you have the endoplasmatic reticulum. So we can study in 3 dimensions how they interact with one another. And here is just another example where we looked at the cytoskeleton, and that is going through the nucleus. something that has never been observed or never been described at least. And we just published this paper where we show that the connections that come out of this filament connect to the nuclear membrane. And it means that the nucleus is mechanically connected to the extracellular matrix. That is something that has been postulated for 30 years, but it has never been shown. And here you see in blue like a donut hole going through the nucleus. And when we actually go back with other techniques and look at many more cells, we actually see that about half of those cells examined showed this phenotype. So that's something that we did not expect to see, and we see it, and then we actually realize it's actually very frequent. And so to come to the question, or to preempt, I guess the question is, how much do we know about cells? Well, we get surprised every single time. There is many more things out there that we don't know. And I encourage people to look at the webpage. You see it right here, ourlab.lbl.gov, and it gives you some more sort of examples of how we use electron microscopy. I think that's all I wanted to say on that. So I'll go back to the, if I can do that. Yeah, I'll go back now and try to stop the screen sharing. If I know how to do that. Here. Okay. So I hope it gives the people sort of an idea of of what this technology can do.
Yes, it was an amazing field trip, like the Magic School Bus books, but through the cells. So this is the real life Magic School Bus here. So
that's actually the funny thing. I always get jealous of like CSI, because they make it look like that was easy, right? As if we knew all that. But that's the same thing as watching Star Trek, and then we're still not going out there where no man has boldly gone before. So I think in some ways I feel like those programs, they're kind of cheating, right? They're sort of giving us sort of cartoon versions of what should be there, but this is sort of real data. This is sort of real cells and real data.
So this is a science nonfiction, we call it.
That's right. That's right. That's reality.
Okay, and so after showing all that, can you then, in pictures, how would you then define the phrase integrated bioimaging?
Yeah, so now I was trying to show you that, first of all, there are multiple techniques that we bring together. And then the question is, how do you integrate that information? If you think about the fate of phone books, right? I would argue that omics is sort of your typical phone book. You have a list of objects that you list in some ways in a database, but nobody looks at phone books anymore. All what we do now is looking at Google Maps. So what is Google Maps? Why is not really anything else but putting multiple types of data on top of one another. And you're doing that sort of at the level of models, right? You have topography to start with, and you put a street grid on top of that, and then you put retail on top of that, and traffic on top of that, and that is the way you search for patterns. And so we need to do the same thing for cells. We're not there yet because it's computationally quite a challenge. The one further problem that we have is there's only one Earth. So no matter what you do, there's only one Earth to map this to. Whereas every biology experiment is kind of its own Earth. So what we really have to do is to take the information that we got from in each data set and abstract it. And one way to do that is to quantify, to measure, to average, and have a prototypic representation of what a particular cell type on a particular experimental condition will look like. And then you will study a number of other cells under different conditions. And again, you will abstract the information through very rigorous quantification. If you then compare the two models with one another, you should be able to say what is different between the cells at two different things. For example, pre-malignant and malignant cells. What are the actual differences? Just knowing that a protein involved in malignancy doesn't really help us all that much. We need to understand precisely how does the organization of a pre-malignant cell differ from a malignant cell. That's one of the themes in my lab.
All right, then. And following up on that question, so what contributions has electron microscopy given to the study of human health?
Well, let's put it that way. For the last 50 years, that's pretty much all the time we had for electron microscopy. It's only been about 50 years. I would say almost everything we think we know about cellular architecture came from electron microscopy. At some point, people were sort of tired of more and more electron microscopy images, and they wanted to see dynamics. And that's why light microscopy has become so powerful, because you can watch things live, whereas in electron microscopy, you cannot really watch anything live because you need to immobilize it in order to image it. But it turns out that a lot of the assumptions that we have, all those little cartoons that we have, that they were fine at the time, but now we're actually realizing that they're not sufficient. So all the people that went away from electron microscopy, a lot of them come back now. And now the real game in town is to combine the information from the varying imaging approaches. And the question is still out there how to do that best. And I would argue that's what this integrated bioimaging initiative sort of comes from, sort of a term that we coined, that is all done best at the level of a model.
All right. Okay. Well, so you also work at, you're director of physical analysis at the Joint Bioenergy Institute.
Right.
One of the things that you guys do there is trying to create new, biofuels or better ways of using our current fuels. And so every high school student learns about this thing called the plant cell wall, right?
Yes.
You learn about it in freshman biology, the more detailed in AP biology. Okay, you're still studying the structure of this cell wall.
We actually have it.
Right. So how much do we know and not know about this textbook topic?
So I can't show it to you because right now, because we're in the process of publishing it. But we actually have determined the structure of plant cell wall. Actually, something that has been a task that has been out there for 80 years. For 80 years, people have sort of been speculating what the plant cell wall looks like. And if you actually look up in Wikipedia or anywhere on the web, plant cell wall model, you will come to a model that has been put out there as a consensus in the field, which it turns out to be completely wrong almost on every single level. And when I actually presented this structure to the field, they all said yeah, you're right. I mean, like seeing is believing. And we not just actually determined the architecture of the plant cell wall, we took that information and we did mechanical simulations. So not only do we know what it looks like, we also understand why it is organized the way it is organized. And the most surprising finding that we had, and in retrospect, it's like is that we thought the plant cell wall is optimized for load bearing, right? It's a cell wall, right? It's a wall, so it's designed for load bearing. Just like in a skyscraper, you think you have a wall and that is for load bearing. It turns out that's not true. It's actually far away from an optimal design for load bearing, but it's an optimal design for flexibility, elasticity, and ductility. And if you think about it, that makes a lot of sense, because if you actually out in a stormy day, the plants, they whip around in the storm like crazy, and they don't break. And they go through enormous deflections from their normal shape, and they don't break. And we think that the organization that we found is exactly designed to have optimized that process. So here's an example where the architecture itself, combined with mechanical simulation, showed us why it's functioning the way it does. And it's an amazing story.
Well, wonderful. We're excited to read that paper when it comes out. I'm sure there'll be cool videos and stuff to go with it too. So yeah, I guess you're right. Seeing is believing. That seems to be a common theme in the history of microscopy, right? From germ theory and everything, when people didn't believe what they couldn't see, and that's what it takes, right, to to prove things, right? So, all right, wonderful. Okay, and so
the fun thing is you can't make images up. Like, it is what it is. You have to deal with it, and you have to find a way to explain it. So many other techniques, while maybe things are a little more like this and more like that, but you have an image, and you have to deal with it.
Absolutely, absolutely. So you also study bacteria and bacterial communities in the soil and in other places. And so, can you tell us what electron microscopy has allowed you to discover and do regarding bacteria?
Yeah. So one of the things that I've been flabbergasted when I first looked at the microbial community first time on the microscope, with the electron microscope. What we found is that in every single space surrounding the cells in the biofilms, there were vesicles. This entire thing was filled with vesicles. And we're thinking, it's very expensive to make vesicles. Why do cells secrete vesicles? By the way, that was a question that had been posed for many years, and people were absolutely resistant. Bacteria do not have the machineries to secrete vesicles, but they are there, and they do secrete them. And we found even more intriguing that they weren't just vesicles; they were chains of vesicles. They were like little pearl, like a necklace with pearls. And they would extend for many microns. We would have hundreds of those little vesicles. We found tubes between that actually connecting bacteria. And so we actually, for fun, we dubbed it the bacterial social network. And when we published it, we got a really huge response. Even the New York Times was actually running the story, actually as a headline on their online version. And they created a little 2.5 minute video that you can look for. It's called Bacterial Private Line. And what we showed is that the bacteria have the ability to communicate in a stealth mode. You can communicate, you can put a message in a bottle and put it out there for other bacteria to pick it up. Or you can have a little torpedo that targets exactly the kind of cell you want to talk to. And nobody else can listen in, because nobody else has access to that message. But more intriguingly, that in some of those vesicles, there are toxins, and they actually kill on contact. And so it's like drones. You never see them coming. And by the time you see them coming, it's too late. And what we think it shows, or we think and we've done a lot of biochemistry and so on to back it up. But the main message that we have is that Myxococcus, the soil bacterium, has an ingenious way to deliver toxic loads to other bacteria. And if you think about it, it had billions of years of encountering other species. You would think that everybody should be immune to everybody. Everybody should be resistant to everybody in the soil. And we think that the reason why it's not, because Myxo delivers its toxic cargo in those vesicles. So that is a fascinating thing, because it never dilutes. It always full strength, it always full power. And I think that is one of the things that our electron microscopy, combined then with genetics and biochemistry and so on, then was able to show.
Amazing. Who would have thought? Okay, have you named the social network? I don't know.
Always going to call them social networks. It's just sort of fun, because the bacteria are always described as social bacteria. They're Myxococcus xanthus. They're social bacteria, so we call them bacterial social networks.
Okay, it's the micro Facebook right there.
Yeah, probably, yeah.
All right, and so what, and you touched about this a bit earlier, but what would you say are the current challenges and future frontiers of electron microscopy?
The sheer amount of data we are able to generate. And so sort of three steps. One is preservation of the specimen in a near-native state. So what we look at is real, and you have to deal with some preparation artifacts and minimize or avoid them. And there are ways to do that by ultra rapidly freezing the sample. And you can do that, and when you do so, then you immobilize your system within a sort of a millisecond. So, it's instant preservation. And then we work up the sample at very low temperature, at sub-freezing temperature, and work them into these resin blocks, at which point the sample is exquisitely preserved. We then cut the sections, and so challenge, then you have to choose which imaging technique you want to use. Transmission electron microscopy, where you go through the specimen, but it can only be very thin. Or you're looking at the block surface, and that's called scanning electron microscopy, and you're just rastering over the block surface. Now, when you combine that with an ion beam that shaves off layer by layer by layer, and as I said, 5 nanometer, which is about 1 protein at the time, you can now build up a 3-dimensional volume where you have image the surface, then you strip away the surface, and image the next surface, strip away the surface, and so on. And you now can look at entire cells, and you can look at entire tissues. And you can do this for a relatively large field of view. So data collection, certainly for any of those data sets, is in order of several days. It can be up to a week or 2. That's just pure data collection. So the challenge then is you have enough access to those instruments, because the instruments are very expensive. They are between $1 and $2 million, and they're not easy to operate and everything. But once you have all that, you have giga to terabytes of data, and we haven't really figured out a good way how to deal with that in a way other than sort of manual. And manual is very tedious. It can take an experienced person several weeks to extract those features from a 3-dimensional volume. So the biggest challenge that we really have is automated ways to extract the information through things like machine learning. So I'm ranging in the type of people that I need to be able to talk to, from biologists to chemists to physicists to computer scientists. And that is really the challenge, because it's such a wide field, which, by the way, brings me to the point is that discipline boundaries sort of disappear. These days, you're no longer a biologist or a physicist. You are everything. And you need to be able to talk to people in all these different fields. Everybody is an expert in their particular field, but the ability for experts to talk to one another requires a very broad exposure to all those different worlds.
And I'd like to add, for our student audience, most of our audience members are high school students and college students, and their parents and college counselors, that what Dr. Auer just described is true, but there's time. It takes time to be able to communicate across all those different disciplines, doesn't it?
And you have no idea how stupid you feel like along the way, because just little things like words that are common knowledge in one field have a completely different meaning in a different field, and just to navigate that world. So I think, if as advice for students, I think just be prepared to feel stupid for a long period of time. Eventually, you'll get it. Eventually, you'll be able to talk to all these people, and eventually, you actually become an expert. People call you an expert, even so that you really feel stupid, and you still often feel stupid at any given time. But it turns out that everybody just knows something, and even if you're stupid in one field, you know a lot about another field. And so in a way, we're all stupid and we're all experts. And so I think that's kind of, one shouldn't get discouraged. By the way, I think in my mind, the very essence of a scientist is you have to have a high tolerance level for frustration, because you mostly will feel frustrated because there's so much to be done and so much to be learned. and you feel very small compared to all these things that are still out there.
Definitely.
But the really funny thing, and I wanna make that point, because I think people actually don't appreciate that, particularly sort of younger people don't appreciate that. I would say 80 to 90% of my day is communication. Being, advocating, writing grants, writing proposals, writing papers. I'm a professional writer, and a professional editor. So for all of you that think that finally, when you go into science, you don't have to worry about languages, you are mistaken. That is actually something you will need more than anything else.
Thank you for sharing that. Well, that leads us well into the next section, in which we talk about your personal training. And so could you describe your academic journey starting from high school?
All right. So first of all, I was born and raised in Germany, and things are a little different in Germany. And hopefully that will be an encouragement for all of you that aren't exactly going to Harvard anytime soon. It almost doesn't matter where you go. What really matters is the passion that you bring for science. So one thing I was very blessed with and lucky with is that I was at a very early stage, about 13, 14 years old, I knew I wanted to be a scientist. I didn't know what that meant, but I knew I wanted to be a scientist. And I was very intrigued by Carl Sagan's Cosmos. That was one of the, obviously one of the big things. And for the longest time, I didn't know what kind of scientist I wanted to be. I thought chemistry was really cool, because that's simple enough to understand sort of at that age. Biology was really not very high on my list. Astrophysics was something I was fascinated by, and it was almost the field I chose for. But then I learned that biochemistry and sort of the inner workings of the cell are even more intriguing. And so I guess my point that I'm trying to make is it doesn't matter what precisely you think you're going to do, because that will change, but a passion to ask questions. So I was well known in my high school to ask what people consider the dumb questions. The teacher just explained it. Why are you asking this question? Didn't he just say that? And later on, the teacher told me, he said, like, those were not dumb questions. They may have seemed dumb, and you weren't quite sure what you were getting at, but you knew that that wasn't the ultimate truth. That was what I was telling you. And so I guess my message to students out there, for those who feel like they have the urge to ask questions and they are told to shut up because the answer is right there, keep asking, because this is all about asking questions. We scientists actually really don't give answers. All we do is we're asking questions and systematically try to work them up into answers. Anybody who tells you that they know, I would be very careful. I think you really have to just ask questions. And it turns out many questions are not that dumb. And if you're actually really thinking about, actually what you actually find in biology anyway is every 5 to 10 years, what was considered the dogma goes out the window. And the one thing you have to watch out for is just to take everything and say like, okay, that is accepted knowledge and this is what I'm going to believe. Challenge everything, want to see the evidence for everything, and keep questioning. And often what you will find is that it's, every scientific discovery doesn't start with a grand plan. It starts with the, that's strange. And that is really what this is all about. So I went to high school. I did very well in high school. It helps to study. But funny enough, I actually hadn't discovered books until I was actually in my undergraduate studies. I just learned everything from just listening to the teacher. And the idea that all this knowledge was in books hadn't actually come to me. It's really silly, but it's really true. And so I came from a family that wasn't very well off, and like books were not a particularly big priority in my family. And so I didn't really know that all this stuff that this teacher was telling me I could have easily read. I was just mesmerized, and I was engaged in class, and I had this urge to ask all these questions, and the teachers were kind enough to respond. And we all inspired by our teachers, and if you have a great science teacher, that will make a huge difference in your life.
Oh, wonderful. And do you have any words? We've had other guests who did their training in high school and undergrad in other countries, particularly Europe, right? You were in Germany, and what was the transition like into being a scientist, in a research institution in America?
Yeah, so let me actually first point out that I went to whatever university would have me. It wasn't a big deal. I really more picked the city where I wanted to live rather than sort of a famous university. Kind of the concept of the reputation of a college really sort of doesn't exist. It's all more or less the same. And I would say just the idea that going to a fancy college that will make your career, or not going to a fancy college will break your career, I think is not true. I think it's all the journey and all what you have in you, and the university just gives you a starting point, and you will always have to sort of work hard to get to the next step and to the next step and to the next step. But that wasn't so important. So what really made my career was a total coincidence that I met somebody that was doing a PhD at the MRC LMB, the Medical Research Council Laboratory of Molecular Biology, the birthplace of structural biology, the birthplace of cell biology, the birthplace of molecular biology. That's where Watson and Crick did the double helix, and Max Perutz did the hemoglobin. And this guy said, like, why don't you come to work, in my lab? I'll arrange something. And I went there, and it blew my mind. Just the ability to have lunch with Max Perutz and him being interested in me and what I was doing, it just blew my mind. And it made me realize that a system where you have flat hierarchies, where everybody is valued the same way, where it doesn't matter whether you're an undergraduate student or a postdoc or a professor or a famous professor or a Nobel laureate, that doesn't matter. That's an amazing place to do science. And the one thing they told me is that if you have the choice between tackling a little problem and a big problem, you should always pick the big problem, because it will take about the same amount of time to solve each and every one of them. But if you solve the little problem, you just solve the little problem. So you should always think of, like, what's the hardest thing to do, and then with passion, go for that. By the way, you will feel inadequate all the time. I mean, that's kind of, it sort of, goes with the territory. I mean, if you feel comfortable, you're not trying hard enough.
Well, that really dovetails well into this question that I ask every one of our guests, right? So it's easy for our students to be inspired by someone like you, but we want them to know that it takes time, right, to attain this level of success, especially because there is a lot of pressure in America to go to a big name school and stuff like that. And so were there times in your academic journey when you felt inadequate or wanted to quit?
Right now, I mean, all the time. You feel inadequate all the time because for some reasons. First of all, you're interacting with very bright people, right? You're interacting with some of the brightest minds in science all over the world, Nobel laureates, other people, and you can't help but feeling inadequate. Everybody who tells you otherwise, they're lying. And I think one of the things that those big schools do, they instill in you this idea that you belong there, that you deserve it, that you are one of the big people. And that's probably a sort of a self-fulfilling prophecy at some point. You know, you just think so great of yourself that you will sort of, you know, succeed in what you do. But I think that often can be counterproductive, because you actually look at who teaches at those fabulous schools, and you will find that often these are not the people you would have thought. You know, those are not people that actually went to Harvard or Yale or Stanford and all of these places. Actually, you will see that at least half of the positions are taken up by people from abroad, with no names, with no big things to point to, and so on. Why? Because they worked so much harder to get to that point. So just because you're at Harvard, it doesn't mean very much. And I think the one big thing about science, and the one thing I love about science, it's the big equalizer. It doesn't matter what family you come from. It doesn't matter how much money you have or your family has. All that matters is whether you can think and whether you're able to work hard. And to comprehend and you know, be open for new ideas. You know, I have to revise my opinions, you know, many times before I hit really the right idea. So you can't be too stuck on thinking you know. And I think one of the things that, one of the counter effects of this great school is that if you stop self-doubting yourself, I think that's not good. I think you always have to self-doubt. You always have to reevaluate and ask the question, am I still doing the right thing? Am I taking all the evidence in? You know, if you're a crime scene investigator, you can't walk into a crime scene and say, looking at, you know, the dead body and say, like, it was the husband. I mean, you have to sort of, you know, take all the evidence in, and you have to be willing to change your mind as evidence sort of presents itself. And not just sort of stubbornly say, like, I know. And so I think it's the same with scientists. You just have to have an open mind, and you have to sort of be willing to abandon your favorite idea, even so it's really your favorite idea. At the same time, you will have to, you can only trust your instincts. You really have to trust your instincts, and you sort of have to go with that. And, you know, there will be times when the entire world will be against you, where what you say is nobody believes, and then only later people realize, well, you were right. And so you have to sort of have a belief that what you say and what you believe in is sort of the right thing. So it's a fine, you know, it's a very delicate walk.
Wow. Thank you for sharing that. And so what advice would you have for current high school and college students who want to pursue a career in your field, say, you know, integrative bioimaging, electron microscopy?
Let me tackle that in a couple of different short ways. One of them is, when I was at the MRC LMB, I asked the director, a very famous person there, said, what advice would you have for a young scientist? And he said, don't settle too soon. So in other words, a lot of people go into research thinking, I want to cure cancer, and then they look out for the cancer lab, and then they want to spend time in the cancer lab, and all they do is just following whatever the field thinks and so on. I think it's much more interesting to pick various different parts of science, because ultimately that will allow you to be original, having had different exposure to different angles. I keep telling my students, it really doesn't matter what project you're working on. Just work on it, do something with it, and you'll learn something from that. And over time, you will have this breadth that will allow you to be original, to bring different fields together. I think that's the one thing I want to point out. I think that a second thing is, find a mentor, find a good mentor. And, you know, and I'm not saying you should all call me, because I'm it's not that I can have so many people join me, but find somebody that you consider a good mentor and get advice at every step along the way. And you will need the mentoring. It's really what I tell students in my lab. That's the most valuable thing they can get from me, is the advice. It's not a stipend. It's not, it's really my guidance. And I think you need a good mentor, and a good mentor will take you a long way. And then the third part is, it does take, at least for a long period of time, hard work to be there on the weekends, be there, But then the thing is, like, if you really love it, it doesn't feel like work. It feels like fun. It feels like you want to be there and you want to do it. And then I would say, trust your instincts. Just trust your instinct. I made many decisions in my life. I once turned down to join the lab of a Nobel laureate, and everybody thought I was crazy, but I didn't feel right to me. And so, and I think I was right in the end. It would have taken me a completely different direction. So just follow your gut instinct. That's really all you have. You will make mistakes, and that's fine, and you will learn from them. And but don't play it safe. If you play everything safe, you will accomplish nothing. So you have to take risks.
Wow, spoken like a scientist. There you go. That's the spirit that brings about discoveries right there. All right. Okay. And so, and a similar question is, what type of background courses would a student need in order to do the research that you do?
Okay, you're gonna hate me for that answer. I'm not a huge fan of courses. I find that courses teach you what was interesting 30 years ago. We still learn about, I'll give you an example. If you look in any microbiology book, right? The vast majority is taken up by genetics, which really is the least interesting aspect of bacteria. And at least that was true 5 years ago. I don't know if it's still true. If you look for bacterial biofilms, microbial communities, bacterial biofilms, you'll find out of a 1,000-page textbook, 1 or 2 pages on bacterial biofilms. But it turns out that 99% of relevance for bacterium is a biofilm. Bacteria actually don't hang out by themselves because they get eaten. Bacteria actually hang out in biofilms. So if you're asking the question, what's the most important part for microbiology? It's biofilms. But nobody teaches you that. The textbooks are 10, 15, 20 years behind. So that's number one. So I happen to think, and you can laugh if you want, I happen to think that the best way to learn science is sort of the Jedi principle. You have a master and you have an apprentice, and you acquire the necessary tools along the way. Yes, it doesn't hurt to read textbook. You need to start somewhere. You need to have some idea what a cell looks like. But you could read any of those, molecular biology of the cell, or any sort of, standard textbooks, and just get an idea of what's out there. And then I think it's really a question of picking a lab and actually doing science. Because the funny thing is, all the lab work at all the universities, and we have UC Berkeley is a great place, but it doesn't prepare you for the real world. And the only way you can get this, as soon as possible, get an internship in a real lab. And it doesn't matter what your role is. It doesn't matter what you do. This is the real world. And all the coursework, as far as I'm concerned, if you want me to say something, math, computer science, English. If you want to be a biologist, I think math, computer science, and English are your 3 most important things to study. Because your world, your world will be a different one than it is right now. It's not about facts anymore. It's not about, learning the facts and reproducing the facts. It's about new things. It's about new directions. It's about, discovering. And you can't discover by reciting textbook knowledge. And that's, in my mind, what courses really do. They just make you recite textbook knowledge. But most importantly, I find them so discouraging, because it is like brilliant people made a brilliant discovery that has nothing to do with me. And it's just off-putting in my mind to read about the success of all the other people. What they should teach in textbooks is the failure. It's the failure that is the most important part. I think somebody should write a textbook of why are we studying mice? Why are we studying rats? Why are we studying Drosophila? That is where the questions are. Like, how do you approach a question? It's not as how brilliant people came up to the brilliant result. It's the process of discovery, and that unfortunately is not really taught in university courses.
All right, there you go. I much.
But I'm a bit of a rebel here, so so take it with a grain of salt.
I think a lot of our our other speakers would agree too, especially in the sciences, right? That ultimately, everyone's advice is to do research, right? It's not to take this class.
Yeah, the real world is, look, this is no different than a barbershop, right? You can't learn how to cut hair by reading a book. You have to do it. And you'll make mistakes, and initially all you get to do is, clean up the hair. But then, you move a little forward, and, they let you do some easy things and so on, and you build up, right? That's the only way you're going to learn. So this idea of a master and an apprentice is really, I think, a good concept. And find a good master. Find somebody who is willing to work with you. Find somebody who is willing to invest time in you, and then pay it back by hard work. Because the people that will invest the time in you, that's a gold mine. You should be so thankful if somebody is willing to take their time and teach you. And don't have an attitude. Just do it. Just learn it. And if it doesn't make sense right now, it will make sense in a while. Have faith in that master that you have chosen carefully will put you on the right path. Take the advice, run with it, do something with that.
Wonderful. All right. Okay, well, let's switch gears to more a sciencey question now. So microscopy involves taking pictures of very small things. Okay, it might be the oldest technique in the life sciences for observing things that are smaller than what the naked eye can see. So the question is, what is it about microscopy that keeps it as a thriving tool and a thriving subject of study, though genetics, molecular biology, and biochemistry have also advanced a lot in the past 100 years? What is it about microscopy that makes it so important and still thriving today?
Well, first of all, like any technique, the technology gets better. So you can do things now that you couldn't do 10 years ago. Like I told you, this focus ion beam, but going through entire cells is something you couldn't do. You will hear that people say that science really, it isn't about new ideas. It's about new technology, because new technology will automatically lead to new ideas. So the technology itself made huge progress, which allows you to do things that you couldn't do otherwise. But why microscopy in the first place? Why am I not doing genomics? Well, the problem is heterogeneity. One of the problems is heterogeneity, and you need to know how things are related to one another. Just to know that California has 20 million people, of which X are Caucasian, Y are African American, and Z are Asian, and whatever else I'm missing now. That doesn't tell you a whole lot how California functions. But if you understand the heterogeneity, why some people settle in certain areas and other people in other areas. That tells you a lot more. So bringing it all together and just saying like, well, it's all the same, that does injustice to the heterogeneity that is out there. And I think human beings are particularly good at making relationships based on visual information. We are visual animals. We detect patterns. And we detect patterns in images much more easily than in other kind of data. And so I would say microscopy is essential to understand all of the parts list and bring them all the parts list together. So it's the unifying thing. It's the one thing that brings all this omics information and all of this other information together. And that's why I think it's very important that we sort of do microscopy.
All right, wonderful. So that's all the questions that I have for you, and we will now go to Alistair, our moderator.
Hey everybody, I'm your moderator for today. Thank you, Dr. R, for your inspiring talk. We do have a couple of questions. The first one is from Sean Lee. He asked, hi, Dr. R, thanks for spending your time with us today. What would you say is the next frontier for integrated bioimaging? Would it be something, as you mentioned during your talk, about computing power to actually process the images your lab is capturing?
It's not so much computer power, I believe, even so that helps. It's more like conceptualizing and abstracting information and representing information, databases, but kind of map-centered databases, biospatial databases. But I think the most important question to answer, I think, is how are tissues organized? That's really the one thing we have very little idea, and how do tissues function? Because at the end of the day, almost every disease manifests itself at the tissue level. It may be caused by certain proteins malfunctioning, but it manifests itself at the tissue level. And that gap from knowing which protein is responsible, but knowing what effect it has on the cell and tissue level, that's the ultimate frontier right now. We do not know, not understand at the macromolecular level tissue organization. And I would say that is the big frontier.
Interesting. Okay. Next question is from Katie. Katie asks, the 3D structure of the nucleus you showed was pretty amazing. What is the most surprising thing you have with electron microscopy?
It's a hard question. It's asking you, like, which of your children do you love the most? I get amazed all the time. It's just like all the things that we take for granted. For example, the bacteria with the vesicles, we didn't expect. The cytoskeleton tunneling through the nucleus is something we didn't expect. You can be sure that the only reason why I haven't been amazed yet is because I haven't looked at it yet. It's almost everything we ever looked at. Like the plant cell wall, when it will come out, you will see it's so beautifully organized, so much more beautifully organized than anybody ever imagined it to be. And in its organization, there is function. And I think we at the very beginning of understanding or making this link between organization and function, particularly at the cell and tissue level. And that is, it's almost anything you look at will amaze you. So I can, and if you, your viewers are worried about that it all will be done by the time they get into it, rest assured, there is so much more that will need to be done. It's a fantastic time now because we laying the groundwork to really ask the fun questions. And that will take another 20, 30, 40, 50 years. And I'm sure there will be more questions after that.
Awesome. And this is actually my personal question. When do you think you're actually going to be publishing that paper? Because I'm actually very curious as well of what that cell wall actually looks like.
It's about this close to submission, so hopefully within a week or two we'll get it underway, and then it will go through review process, which will take probably between weeks and months, hopefully not longer than that. So far the community is on board. They all say it makes a lot of sense, so I hope. You never know with journals, like you just need one reviewer that for whatever reason doesn't like it. But I'm willing to fight this one tooth and nail because it's so beautiful. So I would hope then within half a year it's out and for the public to see.
Gotcha. Awesome. Okay. We have time. I think we have one more question. This is from someone named Tracy. She's a mother. She says, I encourage my kids to explore many different activities. You say it was a coincidence of being invited to work at the lab during your undergrad. But was it because you were actually involved in many different activities that you got invited?
Let me say two things to that. So serendipity is always kind of there are chances in your life that are offered to you, and at that point you need to be ready to take them. Or as they told me, having been invited to work there is one thing, but having been re-invited to work there, that is something that happened because I was putting in the work and I was behaving well, and then they liked what I was doing. But the other thing I want to say is that I totally agree. When I was a kid, I would spend my time playing with Lego, and I was build anything out of Lego, just the basic building blocks. And to this day, I think of a cell as an assembly of Legos. It's all different parts that you put together, and they all sort of work together in a certain way. So yes, all the activities, sports, everything. I mean, you will need this entire thing to sustain you. It's a tough journey. Being a scientist is not easy because it's so much easier to give up. And funny enough, it's not the brightest people that will stay on in science. It's the people that are able to hang on the longest. And people don't drop out because they are not smart enough. Actually, some of the people that were the smartest in my class, they all got stuck and became programmers for SAP or Oracle or those kind of things. It's not about brightness. It's about not giving up. And when life knocks you down, you just dust yourself off and try again. And that is particularly true for science, because in many ways you will be tested, and your frustration level will be tested. The job prospects aren't exactly great, for all the many PhD students that this country puts out there, probably only 1 in 10 or so faculty positions are available. So it's not something, and in the end, it doesn't matter if you have the passion for science. It's not about job prospects. It's not something you calculate. You can't help it. You have to do science. And if you don't feel like you have to do science, maybe you shouldn't. This is something that because you'll be up against people that really, really want this. And so I think it's mostly, it's pure will. It's pure perseverance that will push you through this. And so anything that keeps you healthy and balanced and help you not freak out in life will be an incredible asset for you.
Awesome. Yeah. Okay. That is all the questions we have. Thank you, Dr. Auer. We can actually feel how passionate you are actually when you talk to us. Even though this is a webinar, we can feel the energy through here. So I'm sure our audience members will appreciate this inspiring talk that you gave us today.
Yeah, and I really want them to know this is not something that just started. This has been around for many thousand years now, and there will be many more thousand years to come when we do science. So we need the next generation to come on board, and it's not. If you feel inadequate, you're in the right place.
Gotcha. Awesome.
All right. Yeah. So.
Any closing statements from you, Dave?
No, I just want to thank you, Dr. Auer, for being here, for sharing all your valuable advice. And, I think Alice highlighted what I tried to highlight, ensure that you have a certain presence about you that really is inspiring. And, I think many of our students will now suddenly save up to buy the electron microscopy before they realize it's going to cost $1 million or something like that. You know what I'm saying? So.
Yeah, good luck. If you want to do a crowd funding, let me know, because I can certainly use the money to buy new instrumentation. Thank you so much for having me. It's been great. Thank you.